The third group of macromolecules that we're going to talk about are proteins. Proteins are really important, and we'll talk about them over and over the course of the semester because they're really the workhorses of the cell. And what I mean by that is that they do everything. Like if something is being done, if there's movement, if there's a chemical reaction, if there's a message being sent, if there's something being transported in and out of the cell, it's going to have to involve proteins. And so they really are the one that we're going to talk the most about. Proteins are polymers, and the monomer is the amino acid. There are 20 different amino acids, and they each have the same basic repeating structure. And that is here in the figure from your textbook, and I'll just sort of draw a different one here for simplicity. So this is the amino group, hence amino acid. This is the carboxylic acid or carboxyl group, and that's where the name amino acid comes from. So every amino acid is going to have these two parts. This is the central carbon, so it's in the middle, hence central, right? And then this part here that I've written as R, it's often referred to as the R group or also the variable region. That's the only part that's different between the 20 different amino acids. So this part is exactly the same no matter which amino acid you're talking about. This is the only part that's different. And so this is really the part of the amino acid that's going to make each amino acid unique from the other amino acids. And it's also going to be really important, this part, for determining what the protein is going to look like or what shape it's going to take on. So there's 20 different amino acids, as I mentioned earlier. The amino acids are divided into 4 major groups: non-polar, polar with no charge, and then polar charged with a positive, and polar with a negative charge. And so these kind of categorization is really important because remember that nonpolar means that it's hydrophobic. All of these 3 groups are going to be hydrophilic. So this is going to be important in the shape of the protein. These will have to be away from water. These can be near water. Positive and negative charges, as you remember, can interact in ionic bonds. And so that's why the charges are important to keep track of. So this is one amino acid. This is a second amino acid. When they come together, remember that's a dehydration or condensation reaction, and the bond between 2 amino acids is called a peptide bond. So this is a short chain called a polypeptide. Polypeptide, remember, means many, so many peptide bonds. So in this example, there is 1, 2, 3, 4, 5, 6, 7, 8 amino acids. So notice I counted the R groups. That's just the easiest way to count them. And everything else here in yellow is the repeating part, the part that's always the same regardless of what amino acid is involved. And that yellow part is called the peptide backbone. And again, remember, it's just repeating. It's exactly the same no matter which amino acid is present. The R groups are the part that's different depending on the amino acid. Notice also that the way this chemistry works is that the R groups are always facing in the same direction. So you have one side that has the backbone and one side that has the R group. A couple of other things here. There's a specific directionality to an amino acid chain. So one end has an amino group, so notice there's a nitrogen there. So this is called the amino end or the N-terminus. So terminus just means end. And notice at this end of the molecule, it's the carboxyl group. So this is called the C-terminus or carboxyl end or carboxyl terminus. And so every chain of amino acids or polypeptide is going to start with an amino group and end with the carboxyl group. So it's kind of like the capital letter at the beginning of a sentence and the period at the end of a sentence. Proteins are going to take on specific three-dimensional shapes. Every protein will have a unique shape. So this is a theme that we've been building on. And it's this concept of structure equals function. So every protein has a unique shape. That shape is determined by the amino acid sequence That amino acid sequence is based on the DNA or the genetic material. So really every protein is made from a DNA sequence, and that will determine the amino acid sequence, which will lead to a unique shape for every protein. That shape has to be correct for that protein to function, because structure is going to equal function in biology. So we're going to talk a lot more about the way that proteins take on shape in the next video.